Rat neurons regenerated the mice's brains and restored their sense of smell. Scientists have managed to regenerate damage in mouse brains using neurons grown from rat stem cells. The brains created in this way provide valuable information about the formation of brain tissue and present new possibilities for restoring brain functions lost due to disease or aging processes. When cells of one species grow in the body of another, the resulting organism is called an interspecies chimera. These types of chimeras have been created and used before to study certain tissues. For example, in mice containing cells of the human immune system, to study disease responses. However, creating chimeras for other tissues, such as neural tissue, is more complex. In a recent study, 2 independent teams of scientists showed that adding rat neurons to the brains of mice that had previously had vital parts removed could help regain lost function. The description and results of the research were published in the journal Cell. This study helps demonstrate the brain's potential flexibility to use synthetic neural circuits to restore brain function, says Kristen Baldwin of Columbia University in New York, author of one of the 2 papers. The ability to produce brain tissue from one species in another may help us understand the development and evolution of the brain in different species, says Jun Wu from the University of Texas Southwestern Medical Center in Dallas, co-author of the second publication. Baldwin's team restored mouse olfactory neural circuits, the interconnected neurons in the brain responsible for the sense of smell, using rat stem cells. In turn, Wu's team developed a platform based on CRISPR-Cas9 gene editing technology that can effectively identify genes that drive the development of specific tissues. Mice and rats are 2 separate species that have evolved independently for about 20 to 30 million years. In previous experiments, using a technique called blastocyst complementation, scientists were able to replace the pancreas in mice using rat stem cells. Researchers injected rat stem cells into mouse blastocysts, early-stage embryos that had been genetically modified to incapacitate them from developing into a pancreas. Rat stem cells grew into the missing pancreas and restored its function. Until now, no one has produced brain tissue using stem cells from another species by blastocyst complementation. In the new work, researchers used the platform developed by Wu's team to test 7 different genes and found that knocking out one of them, HESX1, leads to mice without a forebrain, a large area in the brain that coordinates most of the animal's behavior. These mice died quite quickly. However, when the researchers injected rat stem cells into the blastocysts of mice lacking HESX1, the rat cells filled the niche, creating the mouse forebrain. Rats have larger brains than mice, but the rat-derived forebrain developed in the mouse brain at the same rate and size as the natural mouse forebrain. Moreover, rat neurons were able to transmit signals to neighboring mouse neurons and vice versa. The researchers did not test whether forebrains derived from rat stem cells changed the mice's behavior. There are no good behavioral tests to distinguish rats from mice, but our experiment shows that mice with rat forebrains do not behave unusually, Wu says. In the second study, Baldwin's team used specific genes to silence or remove neurons responsible for the sense of smell in mice. Scientists indicate that by silencing genes, they aim to create a model that imitates what is observed in diseases characterized by neurodevelopmental disorders in which some neurons have problems communicating with the environment. However, by removing genes, they simulated some degenerative diseases. The resulting adult animals had disrupted circuits connecting olfactory neurons in their noses to areas in their brains, preventing them from using their sense of smell to find the treats the researchers had hidden. The researchers returned to the blastocysts and injected them with rat stem cells. As the animals grew older, scientists monitored the neuronal activity. They found that the mice had functional olfactory neural pathways consisting of both rat and mouse cells. These cells could communicate directly with each other. Rat stem cells restored mice's sense of smell, but in different ways depending on the model. In this case, when specific genes were silenced, mouse neurons were present but inactive. Neurons created from rat stem cells created better organized brain areas compared to a model in which genes were removed. But when the chimera was tested to find a hidden treat, the model in which the genes had been removed performed better. This suggests that adding replacement neurons is not a plug-and-play solution. If you want a functional replacement, you may need to empty out the dysfunctional neurons that are just sitting there, which can be the case with some neurodegenerative diseases as well as some neurodevelopmental disorders such as autism and schizophrenia, Baldwin explains. This truly surprising result allows us to look at the differences between these two disease models and try to identify mechanisms that could help restore function in both types, he adds. Currently, as part of clinical trials, scientists are transplanting stem cells and neurons into people suffering from Parkinson's disease and epilepsy. But we don't really understand how well it will work. Thanks to our brain models, we can start getting answers at a faster rate. This study provides a system in which we can evaluate the feasibility of conspecific brain complementation on a much larger scale than a clinical trial, says Baldwin. Blastocyst replenishment is still a long way from clinical use in humans, but both studies suggest that stem cells from different species can synchronize their development with the host brain. Wu and Baldwin say their research addresses long-standing concerns about the development of chimeras, particularly for transplanting tissues or organs from animals such as pigs to humans. In addition to ethical considerations, there is a fear that the human body will reject the transplanted organ. Because the teams added rat cells so early in mouse development, long before the immune system was established in embryos, the animals' bodies never learned to recognize these cells as foreign and never attacked them. thought-controlled exoskeleton allows a paralyzed person to walk. Thanks to a brain-controlled exoskeleton, a Frenchman who had been paralyzed for 4 years was back on his feet. The exoskeleton, weighing over 60 kilograms, gives hope to paralyzed people to regain the ability to move. 28-year-old Thibault lost the use of his limbs after falling from a great height. After many months of training and work by French scientists, he was able to get back on his feet. He was helped in this by an exoskeleton and sensors implanted in the brain, thanks to which the man could control the device. The scientists behind the device say that while it has the potential to improve patients' quality of life and autonomy, it will be years before the exoskeleton is commercially available. Many improvements are still needed, and the exoskeleton should be made lighter to make it weigh less. About 20% of the cervical spinal cord is damaged. Cases end with tetraplegia, i.e. four-limb paralysis. This is the most severe injury of its kind. The brain is still able to generate commands that would normally move the arms and legs, but there is simply no point in following him, said Arlem Louis Benabid of Université Grenoble-Alpes, lead author of the study published in The Lancet Neurology. A team of experts from the hospital in Grenoble-Alpes, the biomedical company Clinatech and the CEA Research Center qualified Thibault. The man did not want to reveal his surname for the experimental program. The Frenchman was implanted with 2 electrodes, but unlike other operations of this type, the sensors were placed not directly in the brain, but on the outermost membrane that protects the brain, the so-called dura mater. Researchers also scanned Thibault's brain so they could map during exercise which areas became active when he thought about walking or moving his arms. The exercises involved controlling video game characters using signals sent from the brain. Only after Thibault had achieved satisfactory results was he allowed to switch to the exoskeleton. The implants implanted between the brain and the skin of the skull contain 64 electrodes. They are placed on either side of French's head to pick up signals from the motor cortex. The implants communicate wirelessly with the computer. During 2 years of training in video games, the artificial intelligence algorithms used in the device learned to recognize the signals sent by the 28-year-old's brain when he tried to move the avatar. Each decoder transmits brain signals that are then converted by an algorithm into the movements the patient was thinking about. It is this system that sends physical commands that the exoskeleton executes. The 28-year-old, strapped to the exoskeleton, initially made clumsy movements, but over time he learned to walk and, for a video showing the progress, he walked 10 meters and performed a series of complex hand movements using the futuristic machine. I felt like I was the first man on the moon, Thibault said. I didn't move for 2 years and I forgot what it was like to stand, he added. To date, much of the research into improving the mobility of paralyzed people has focused on electrical muscle stimulation using machine-brain interfaces. The exoskeleton, however, takes a completely different approach, bypassing the body entirely. While trying to walk, Thibault, who spent 2 years in the hospital, was able to activate the machine with his brain 7 out of 10 times. He performed 70% in the tests. Commands given to him by the researchers. As he admitted, the greatest difficulty for him was mastering the movements of his wrists, due to the greater dexterity required. For safety, the exoskeleton was strapped to the ceiling. But scientists are working on a machine that will maintain its own balance. We need more computing power. If the patient could count on the stabilization of the exoskeleton, walking would be much easier, Benabid said. TiVo implants are still working 27 months after implantation. Scientists now want to invite 3 more people to the program.